Alright guys, so what we're going to be talking about today is gel electrophoresis. This video is going to focus on how to set up a gel electrophoresis apparatus and how to do it correctly and accurately so that your gel will run. And so we're going to be running our gel electrophoresis today with fish proteins from different species of fish. And then we are also going to be using what we call an SDS gel, which comes in a box that looks like this. So the first thing we want to do is that we want to take one of these little packets out, make sure that they're sealed, and if they are, we want to make sure we open it and take out a casing that looks like this. You can't really see it, but there is a gel inside of that casing. And so the next thing we want to do is fit the casing onto the gel electrophoresis machine. And so the main things to remember in here is that we want to make sure that the bottom of the casing of the gel as well as part of the apparatus are completely flush together, and that we make sure that we are pressing it down so that there's a seal being made between the gel tray or the gel casing, sorry, and those little rubber pieces that you see in the video. So what you see here is that I'm pressing it down. I'm making sure that those edges right here are flushed. Okay, we're creating a nice little seal. And then just to make sure that everything is sealed and basically fitting firmly, we want to make sure that we clip down the casing, and then once we clip it down, we want to go ahead and insert it into our tray, and it should look something like that. The next thing we want to do is, you'll see here is what I'm holding is TGS buffer. We want to make sure that we take this and we pour it into our gel tray. Or a gel electrophoresis tray, and we want to make sure that it's all the way to the top. And then after that, we want to very carefully start pouring it into the gel casing. And so what we want to do here is we basically want to submerge the whole entire gel, but more importantly, there's going to be little wells up at the top where we insert our sample later on, and we want to make sure that those are submerged. And so you'll see me here carefully pouring it, and part of this is making sure that the liquid that's here inside of the gel casing and then also in the back, the levels should be staying consistent. If it drops at any point, it means that we don't have a perfect seal, and so we have to take it apart and redo it. But as you can see, my levels are staying steady, so I just keep pouring until I basically get to the very top and make sure that my wells are submerged. So once I've done that, it should look something like this. I've got my gel submerged, I've got my clips, I have my buffer that's all the way to the top of the bottom of the container. And so I'm ready now to start putting in my sample. And so if we look at our lab handout for today, normally what we would do is every 2 groups would get 1 gel. And so we would have group 1 have their samples right here, and group 2 have their samples right here. And in between those 2 samples, there would be what we call ladders or standards. And one would be a kaleidoscope, and one would be actin and myosin. And so the reason we need these is because once our gel runs, we need something to compare it to. And so what these do is that they show us where certain proteins are located on our gel. And so when we run it, wherever those bands are, we are able to compare it to our actual samples, and we're able to tell what that band is, which is very important. And so in today's lab, each of our samples will be a different fish. So we have, for example, we'll probably have salmon, squid, mackerel, sardines, and I believe the last one was tilapia. And so to do this, it's going to end up looking something like this. And so the very first thing that we are going to need for this process is a pipette mitt. And we're going to be putting, that was kind of quick, but we're going to be putting 17 microliters into each well. And what you'll see here is that I need a very specific tip for this, and we call it a protein gel loading tip. And this tip is a little different than the rest because it's more flexible. And so I'm able to basically run that tip along the edge so that I can hold it steadier. And so what we're doing here is we're taking the kaleidoscope and I'm going to insert it into the gel. And you'll see that I'm using 2 hands because I need to be able to steady myself. This is probably the hardest part of this lab, right? Because we want to make sure that our tip is in the well nicely so that our sample doesn't run everywhere, but at the same time we don't want to insert the tip too far down because then we'll puncture the well. And if we puncture the well, then our sample's just going to go everywhere and it's not going to run properly. And so using 2 hands and looking at the top, you'll see that I put my gel in right there. And I didn't stick it far enough that it would puncture anything, so that's nice, and it's sitting nicely on the bottom. This is my actin and myosin standard, and so again, I'm doing the same thing. I've changed my tip, and I'm now going to insert it into the well with 2 hands to steady myself, and I'm going to carefully insert it, and as you see, it's all sinking to the bottom nicely, and it's not running everywhere. After this, I'm going to do the same thing for each of my samples of fish, but just for the sake of time, we're just going to show you one. Again, inserting it nicely so that it's in the well, but not so far that I'm going to puncture it. And so as you see, it's running down into my well nicely, and it's not spreading everywhere else. So then after this, you should have something that looks something along those lines, but each of the wells would be full. So my next step is going to be to put my lid. And what's important about this is that I need to make sure that my red electrode goes to the red one and my black goes to black. Okay, so we really need to make sure that those colors match. And once I've done that, I can carefully go ahead and start putting my lid down, make a nice little seal. And then same thing, I want to make sure that red goes to red, black goes to black. And so after that, I'm going to set my voltage to 70, and this is pretty easy. You just press the power button, adjust your voltage, and then press start. And after I do that, I'm going to let it run for 45 minutes. Sorry, this was me setting my voltage. And luckily, one of our TAs, Melissa, one of her students from previous semester, actually made a time-lapse video of her gel running. And so this is what it looks like in a fast-forward situation. And as you can see, we have our ladder right here that we're going to use to compare it, our own values to. All right, so once I'm done, I'm going to turn off my machine. I'm going to remove the lid. And I'm going to make sure I unplug everything, even though I've turned off the power. I'm going to carefully take out my gel, unclip it, and remove everything. Make sure that when you are taking the casing off, that you're doing it over the tray because all that liquid is going to come out. Perfect. Right, and then I can just set it to the side. And then here's probably another really tricky part of this lab. So for the next part that we're going to do, we're going to dye our gel so we can actually see those bands. But in order to do that, we need to take the gel out of the casing. And as you can imagine, it's pretty tightly put in there. We want to make sure that we're as careful as possible to not rip it. If it rips is okay, but it's just not ideal. It's not as pretty. And so as you can see, I took off my gel. I used a razor to get the edges going. And then after that, I just carefully used my finger to scoot out the gel. I then went ahead and put it in a Tupperware, and we're going to rinse all that buffer off. And so I'm going to rinse it 3 times with DI water. Here's me taking out that gel very carefully. I think this is the first time that I've not ripped it, so I was pretty proud of myself. So we're going to go ahead and rinse it out with DI water, get all that buffer off. We're going to do that 3 times, really making sure to get it as clean as possible. And then we're going to go ahead and take our dye. And so what we do in this situation is the gel is still in our Tupperware. We're going to put in 70 milliliters of our dye. Make sure that our gel is submerged. Then we're going to put the lid on, and we're going to take it to a shake table. And so we're going to go ahead and make sure that our Tupperware with our gel and our dye are very firmly secured to the table. Then we're just going to turn it on. And after we've let it sit in there overnight, we're going to go ahead and take out the dye. So the dye can just go back in the bottle because we can use it several times, which is nice because we can reuse it for multiple sections. And so then after that, what I want to do is reseal it up, make sure nothing's going to get spilt, and I'm going to take it back to the sink and rinse it with DI water 3 more times to get as much of the dye as I possibly can off. Then we're going to go ahead and photograph our gel. And so normally the way we would do this is that we would place the gel right here onto this light box. And then we would put the lid on top and turn on the light and be able to take a really clear picture of our gel. And this allows us to just see it more clearly. So this is what the apparatus would look like when we put it all together. And then that would be an example of one of the gels from the previous semester. There's another picture. And when you guys do a lab report for this class, right, what you would normally want to do, especially if this is the type of process that you are doing in your research, is you would want to have an example of your gel, a picture, and then make sure that you clearly label each row so that the people that are reading your report are able to tell what your what you were looking at, and it just makes it more clear. You also want to label which bands you can find across each sample. So this would be a myosin heavy chain right around here. And we know that because if we look at our actin-myosin ladder or our standard, right here is where we would find that. And so we're able to tell that these samples all have myosin heavy chain. So that's it for today and how to set up a gel electrophoresis. We're also going to talk about what exactly goes on during a gel electrophoresis, the science behind it, but that'll be another video. And if there's any confusion, always feel free to email any of your TAs or your lecturer.